Hi. So this class we are continuing with the various methods to overcome the intraocular barriers. So today this is the third session. So today we are discussing on the use of the drug carrier systems to enhances the permeation or to overcome the intraocular barriers. So these were the three main methods to overcome the intraocular barriers. Includes the penetration enhancers, which we discussed the first class, and use of mucoadhesive polymers we discussed on the last class, and the third session we are discussing on the drug carrier system. The various drug carrier systems used to overcome the barriers are use of nanomicelles, nanoparticles, nanosuspensions, liposomes, dendrimers, in situ gelling systems, contact lens, implants, and micro needles. Let us discuss one by one. So nanomicelles. So micelles, you know what are micelles. So this micelles in the nanoparticle size, we call it as the nanomicelles, which are most commonly used carrier systems to deliver the drug molecules. These nanomicelles are developed by using the amphophilic molecules, either surfactants or the polymers. The nanomicelles offers the higher encapsulation capacity. What is meant by higher encapsulation capacity? So you have seen the structure of the micelle. It is spherical in shape and it is having certain cavities. So the encapsulation efficacy explains how much amount of drug can be incorporated inside these vesicles. How much amount of drug can be incorporated into this micelles. So this nanomicelles offers, or it can carry as a carrier system, this nanomicelles can carry higher amount of drug within this micelles. That means even if you administer a small volume of micelle also, this can lead to administer higher amount of drug into the ocular cavity. Then second advantage it offers is it is easy for preparation. The particle size of the micelles are very less. As a result, it enhances the bioavailability and therapeutic efficacy of the drug molecules. So this advantages offers a greater therapeutic outcome for this nanomicelles. So first thing, it carries higher amount of drug. It is easy for preparation. It is small, size is very small, and it offers a higher therapeutic or bioavailability of the drug molecules. The second one is general nanoparticle. In the case of nanomicelles, the particle size was nano in range and it was in the shape of the micelles. Whereas the nanoparticles, they are purely 20,000 nanometer, the maximum range. So here the major advantage is the particle size is very less. It may be spherical in shape, but particle size are very small in size. So this can be prepared by using the lipids, proteins, or the natural or the synthetic polymers, which includes albumin, sodium alginate, chitosan, and all those polylactic or PLA and polycaprolactone, etc. These nanoparticles can be of two types. Generally, they are of nanocapsules as well as nanospheres. Nanocapsules as well as nanospheres. By meaning of nanocapsule or nanosphere, it is doesn't mean that nanocapsule are capsules in shape and nanospheres are spherical in shape. Both the nanocapsules as well as nanospheres are spherical or round in shape almost, but it differs in their content. In case of the nanocapsule, The drug is enclosed inside a polymeric shell. Enclosed inside a polymeric shell. You remember we learned the reservoir system and the matrix system in the controlled drug delivery. What was reservoir system? That is, the drug is incorporated and it has been covered with a polymeric matrix. So inside it is a reservoir system, and in the case of matrix system it is a hybrid system where the drug as well as the polymers are mixed together and it is being prepared. That was a matrix system. So the same concept applies for the nano capsules as well as for the nano spheres also. In case of the nano capsules, the drug is enclosed inside the polymeric shell. So outside shell is there, inside the drug molecules has been incorporated, whereas in the case of nanospheres it is being developed with a polymeric matrix, and the drug molecules are incorporated inside this polymeric matrix. So that is the nanospheres. Okay, so whether for the past decades many researchers has been working on these nanospheres and nanoparticles, considering the various advantage of these nanoparticles for the delivery of the drug for the ocular drug delivery, especially to deliver the drug molecule especially into the anterior as well as the posterior ocular tissues. So the major advantage, why they become the promising candidate, the major advantage they offers is small in size, then less irritation and sustained release property, which helps in avoiding the frequency of administration. As we know, if something, a small particle, if it goes into the eye also, it will be very much irritating for us. So again, the size of these nanoparticles is very less. It never produces any sort of irritation after administration of the drug into the ocular cavity. At the same time, this produces a sustained release. That means it slowly releases the drug for a longer period of time, which will help in reducing the frequency of administration. And one of the main drawback of this, like the aqueous solutions we have seen, the aqueous solutions if you administer, either it mixes up with the tear and it will goes out to the puncta into the nasal cavities. So a major portion of the drug may be spoiled or maybe thrown out of the ocular cavity if you administer as an aqueous solutions. The same drawback also exists in the case of nanoparticles also, because nanoparticles if we administer as a solution, the solution or the suspension that contains the nanoparticle, what happens, even though it is small in size, it favors various advantages also. Because of the tear dilution, there are chances it may be spoiled out, it may be drained out of the ocular cavity. So this issue can be overcome by developing it with the help of the mucoadhesive polymers. By incorporating these nanoparticles in the mucoadhesive polymers, these polymers will adhere into the cornea. It will adhere into the precorneal pockets, the cul-de-sac. So this adherence by this mucoadhesive polymers, and these polymers will slowly releases our drug molecule, the nanoparticle, the nano-sized drug molecules, and these drug molecules can easily permeate the cornea and it can reach to the anterior and the posterior portion of the ocular cavity. Then, to develop this mucoadhesive polymers, to develop the mucoadhesive nanosystems, the various polymers used like polyethylene glycol, chitosan, hyaluronic acids are majorly used. This we have discussed in the last class. And the chitosan coating is the most widely exposed one because of its residence time. The chitosan is generally the positively charged and the corneal sulfurs is negative in charge. So these are the oppositively charged molecules. The chitosan gives a positive charge and the corneal cells is of negative in charge, and these oppositively charged molecules always attract each other. So the formulation developed with using the chitosan retains in the cornea for a longer period of time. The next one is the nano suspensions. So these nano suspensions are colloidal dispersions of submicron drug particles. They are stabilized or developed by using the polymers or surfactants. So this is also a promising strategy for the delivery of the hydrophobic drug molecules. What are the various advantages this nano suspensions offers? Nano suspensions are easy for sterilization, then ease of hydro preparation or formulation, less irritation, higher residence time, and it enhances the ocular bioavailability of the drug molecules. As we know, the suspension itself having a higher viscous thing, and the nano-sized particles are being suspended into this. So because of this system, each offers a higher bioavailability, a less irritation, and a higher residence time. An example of this nano suspension study has been conducted as glucocorticoids such as like prednisolone, dexamethasone, hydrocortisone as what are some of the drugs which has been highly recommended and treated for treating the inflammatory conditions in the anterior segment of our eye. Okay, so the continuous therapy of these drug molecules can lead to formation of cataract formation, glaucoma, as well as the damage of the optic nerve. So these drug molecules has been developed as a nano suspensions which found with these drawbacks like cataract formation, glaucoma, etc. can be overcome by developing or by formulating these glucocorticoids as the nano suspension. How it overcomes? See, if you are directly administering as a solution of this glucocorticoids, since there is a large drainage of these drug molecules can happen, and thus a large amount of drug need to be administered, and we couldn't find out how much of drug will be permeated into the anterior and the posterior segments. It varies from people to people. It varies depends upon the drainage system and how the patient is administering the drug. Are they closing the puncta? So some people they will get a huge amount of drug, some people they will get a very less amount of the drug. And again, to cure the inflammatory conditions, there may be continuous presence of the drug molecule also might be required. So we have a less control of the amount of drug being entered into our eye if you are administering as a general solution. So this drawback can be overcome if you made it as a nano suspension, because it ensures the maximum amount of drug. If you administer one microgram of a drug molecule, it assures that a 90% of drug can crosses the precorneal barriers and it can enter into the various anterior and the posterior segments of the ocular. So developing this into the nano suspensions, we need to administer only a less volume. That less volume will help in treating the inflammatory conditions, and since it's only a less amount of drug, there is no chances of formation of the cataract or glaucoma, or it never damages the optic nerve. Okay, so this type of nano suspensions can help in development or delivery of this glucocorticoids. Another formulation is the liposomes. So these molecules like nanoparticles, liposomes, everything in detail you will be learning in another chapter, another segment. We'll be learning in detail how to prepare, what are the characteristics of this molecule. Okay, so there you will get the more clarity on development or preparation of this drug delivery systems. Again, the liposomes is also another novel drug delivery systems which are developed. It's not only for administration into the ocular cavity, but it can be administered to any route, to any parts of the body. So all these drug delivery systems are general novel carrier drug delivery system, not only for ocular but for all parts of our body. It can be all routes can be administered, but here we are learning especially the liposomes for the ocular drug delivery. So these liposomes are the lipid vesicles with one or more phospholipid bilayers enclosing an aqueous core. So if you look into this picture, you can understand that there is a hydrophobic tail, and it's a hydrophilic head. So as it is mentioned, it is made up of phospholipids which is having a phobic tail as well as a hydrophilic head. So these phospholipids are having both hydrophilic as well as a hydrophobic in nature. And this forms a vesicles in nature. Vesicles, it's like a micellar structures it forms, and this vesicles, inside this vesicles we can incorporate certain amount or certain quantum of the drug molecules can be incorporated. So the size of these liposomes varies from 0.08 to 10 micrometer based upon the phospholipids. Okay, and these liposomes are classified as small unilamellar, large unilamellar, and multilamellar vesicles. Okay, so unilamellar means this carries only one bilayer. Okay, where the one layer bilayer itself they are having in the size range of small, which is ranging from 10 to 100 nanometer, and large unilamellar ranging from 100 to 300 nanometers, etc. So what the advantage these liposomes offers? The higher biocompatibility, it's having a cell membrane like structures, and it can ability to incorporate both hydrophilic as well as the hydrophobic drugs. So the biocompatibility or the compatibility of this liposomes made of phospholipids with the ocular cavity itself is one of the main advantage, that it will not produce any eye irritation or redness to the eye. So compared with that of the nanoparticles, the size of liposomes will be a little bit higher than that of the nanoparticles. But since these liposomes are also been made of lipophobic molecules, which offers, and it can carry both hydrophilic as well as hydrophobic drug molecules, and it offers a higher permeation of the drug molecules. So, and these liposomes are effective for the delivery of drug towards both anterior and the posterior segment. The positively charged liposomes. The liposomes are of positively charged, and which can again bind with the negatively charged corneal surface, which lead to increases the retention time and increase in the residence time of your drug molecule. The same principle what we learn in the chitosan. The next one is the dendrimers. So dendrimers are characterized as the nano size, because first thing is they are nano in size. Second thing is they are highly branched, and they are having star-shaped polymeric system. So this is the picture of a dendrimers. Okay, so this main characteristics of the polymers is they are like branched chain they forms, and they are of nano in range. So these branched polymers, they are available at a different molecular weight, and they are having certain terminal amine hydroxyl as well as carboxyl functional group. So this terminal hydroxyl and carboxy functional group plays a major role in targeting the drug delivery, because this dendrimers carries the drug molecule. The terminal tail portion of this dendrimers can bind with the corneal cells, and it can adhere to the corneal cells. That means this dendrimers carries the drug molecule, it binds with the corneal cells, and it slowly delivers the drug into the eye. The highly branched structure of dendrimers allow incorporation of wide range of drug molecules irrespective of their nature, which can include both hydrophobic as well as hydrophilic drug molecules. Next is in situ gelling system. So this in situ, the word itself means that is the gel formation happens in the site. In situ means in the site. So we are administering the drug into the site of eye, that is into the ocular cavity. So once the drug is being administered into the ocular cavity, we are administering the drug as a solution, a less viscous solution. But once after administering into the ocular cavity, based upon the certain characteristics of the polymer and characteristics on the ocular cavity, this solution will be immediately converted as a gel-like consistency. So while administering, it is a solution only, but after reaching into the ocular cavity, it get converts to the gel form. So that we call it as the in situ gel formation or gelling systems. So this type of hydrogels polymeric solution which undergo solution gel phase transition from viscoelastic gel in response to environmental stimuli, because based upon the environmental stimuli or environmental condition in the sense the condition of our eye and the condition of the polymers what we used. So based upon this environmental conditions, there is conversion from solution into the gel. So if you look into this picture, we are administering as an eye drop which contain the drug. Which after reaching into the cornea, based upon the pH change, because corneal pH and corneal temperature, this change, due to this change in the pH and the temperature, that's what I mentioned earlier, based upon the environmental factors, that is environment of the eye and the conditions of the polymers. All polymers will not change based upon the pH. All polymers will not be changed based upon the temperature. It depends upon the condition of the polymers also. So while administering it as a solvent, it's as liquid, and after reaching it becomes a gel and it will adhere to the cornea and it releases the drug molecule. The gelation is based upon the temperature, pH, ions, or even by UV radiation. But the most important one and easy is the temperature as well as the pH. So many studies has been conducted or still is going on by developing in situ gels based upon the pH as well as temperature. And several gelling polymers used like the polaxamose, then multi-block copolymers made of polycaprolactone, polyethylene glycol, lactates, glycolides, then polynisopropyl acrylamide and chitosan. And these polymers form temperature dependent micelles and aggregates which gellify after further temperature due to aggregation or packing. This is what we have explained. For this drug delivery, these polymers are mixed with the drug in the solution state and the solution can be administered, which forms an in situ gel depot at the physiological temperature. That's it. Next one is the contact lenses. So contact lenses, as you know, which is an alternative for the spectacles or what we are using. But this type of contact lenses can also be utilized as a drug delivery systems. So how this contact lenses work? So these contact lenses are thin curve-shaped plastic discs which are designed to cover the cornea. Now, after application, the contact lens adheres as a film of the tears over the cornea due to the surface tension. It's attached properly with the cornea. And the drug-loaded contact lens has been developed for the ocular delivery of numerous drugs such as beta blockers, antihistamines, and antimicrobials. Now, how does this work? And this contact lenses offers a longer residence time, higher drug influx to the cornea, and less drug inflow into the nasolacrimal duct. That is, loss of drug is reduced, higher residence time it offers, and there is higher movement of the drug molecule. So how the drug is being incorporated into this lens? It's a simple thing that this lens can be soaked into the solution that containing the drug. The lens can be soaked into the solution that containing the drug. So this lens can take up this drug molecule. Once you keep the lens into the ocular cavity, the drug molecule will move from the lens into the ocules, into the ocular cavities, into the cornea. So this way, this contact lens acts as a depot, as a reservoir for the delivery of the drug molecules into the system. The next one is the implants. As we know, implants are something like some devices which has been inserted into the body cavity by certain surgical or non-surgical methods. Especially, but they are majorly inserted as by surgical procedures. Okay, so this insertion of the implant by means of certain surgical procedures which offers the localized as well as controlled drug release, and which can overcome certain drawbacks of injections and associated complications of this injections, and they are placed intravitreally by making an incision through sudden surgery. Okay, so this offers the sustained release, local drug release, reduced side effects, and ability to circumvent the blood retinal barriers. So it offers the sustained drug release, the local drug release, and it reduces the side effects, and it also helps in the higher permeation of the retina. The last one is the micro needles. So micro needles based technique is at its most emerging one to the delivery of the drug into the posterior cavity. So if you look into this, these are micro range type needles, though these needles which carries or which is same as that of the drugs, and because of its smaller size, it can easily be administered into the site of action, and this will be detached from the device. So if you look into the picture, you can see the drug slowly undergo dissolution, it undergo deformation, and the tip is being separated. So this is the micro needle tip. We can see it can slowly administered into the cornea, and the tip which contain the drug will be separated out of the system, and it will slowly releases the drug from this micro needles. And these techniques provide an efficient treatment strategy, especially for the vision threatening posterior ocular diseases like macular degeneration, diabetic retinopathy, and the posterior uveitis. So for this sort of diseases, which are of various vision threatening diseases, the micro needles seems to be highly efficient, and it reduces the risk and complications associated with the intravitreal injections. Next thing is, it also helps to overcome the blood retinal barriers, which can customized to penetrate the main barriers of the sclera as well as cornea, and these needles also helps to deposit the drug or the carrier system into the sclera through the narrow space between the sclera and the cornea called as suprachoroidal spaces. So through this suprachoroidal spaces, the micro needles can be inserted directly into the scleral portions, so that it can deliver the drug properly into the sclera, or this also act as the carrier into the delivery of the drug into the interior portions. Okay, so that's it. So thank you all. So listen and study. Okay, bye bye.